Welcome to the deep dive. Today we are taking on, well, one of biology's most complex and frankly brilliant systems, your internal defensive architecture, the immune system.
It really is. Our mission today is to do a deep synthesis, really, of the foundational strategies outlined in Chapter 43 of Campbell Biology. We're going to try and go beyond just definitions and explore the amazing molecular machinery behind how your body fights everything off.
And it's just staggering in its complexity, but the whole thing, it really boils down to one simple question, doesn't it? How does the body tell self from non-self?
Exactly.
There.
That is the central question. If you get that wrong, it's catastrophic.
Right, if you mistake a friend for a foe, you get autoimmune disease.
And if you fail to spot the enemy, the whole system is compromised. So evolution has sort of settled on these two brilliant interconnected strategies. First, you have the rapid generalized defense that's innate immunity.
The one all animals have.
The one all animals have. And then you have the second strategy, which is slower but highly specific and, crucially, it remembers. That's adaptive immunity, found only in vertebrates.
Okay, let's start there. With the rapid response team, the innate system, I always think of this as like the uniformed police force and the border patrol.
That's a great analogy. They don't need a specific ID. They just recognize broad danger signs.
So where does it all begin?
Well, it begins with the most basic line of defense. Physical barriers. Your skin, for instance, isn't just a covering.
Right.
It's an acidic environment, usually around pH 3 to 5, which actively stops bacteria from growing.
And then in the places where we're open to the world, like our airways, they're lined with mucous membranes.
Which produce this thick, viscous mucus that's basically a physical trap for anything you inhale or ingest. And if pathogens get past that, there are chemical barriers waiting.
Like lysozyme, in tears and saliva.
Yep, that breaks down bacterial cell walls, and if a microbe makes it all the way to your stomach, it faces an acid bath. We're talking a pH of 2. It's lethal to most things.
But, barriers get breached, a small cut, a splinter. So once the fight moves inside, we're relying on cellular defenses, starting with the hungry ones, the phagocytic cells.
Right. Phagocytosis literally means cell eating. We're talking about macrophages, the big eaters, and the highly mobile neutrophils that circulate in the blood.
And they literally just eat the invaders.
They engulf them. They extend these things called pseudopodia, wrap around the pathogen, and pull it inside into a little bubble of vacuole.
So they trap it. But then what? Do they just hold it there?
No, they fuse that vacuole with a lysosome. And this is where the action happens. The lysosome is, it's basically a bag of digestive enzymes and toxic compounds.
A sophisticated acid bath.
A very sophisticated acid bath. The pathogen is completely destroyed, and the leftover debris just gets released back outside.
What I find fascinating is how they know what to eat.
Yeah.
If they don't recognize specific pathogens, how do they tell a threat from just, random debris?
That is the genius of it. It's all about pattern recognition. They use these receptors called toll-like receptors, or TLRs.
TLRs.
Okay, and they don't look for a unique ID badge. They look for generic signature traits, things that are common to whole groups of pathogens, but are completely absent from our own cells.
So it's like recognizing a standard issue uniform that only the enemy wears.
Precisely. For example, TLR4 recognizes lipopolysaccharide, which is a key part of many bacterial cell walls. TLR3 is inside the cell, and it recognizes double-stranded RNA.
Which is a huge red flag for a virus.
A huge red flag that a virus is replicating inside. It's a general alarm system that's built for speed, not subtlety.
And beyond these main phagocytes, there are other specialized units, aren't there?
There are. You have eosinophils, which are really important against bigger threats like parasitic worms.
Yeah.
And then you have the natural killer cells.
The assassins.
The assassins of the innate system, yeah. They don't eat anything. They just patrol, looking for any of your own cells that look stressed or abnormal, like they're infected by a virus or have become cancerous.
And they just kill them.
They induce them to commit suicide, a process called apoptosis.
Which brings us to something we've all experienced. Inflammation. That redness, the swelling, the heat around a cut.
That is the local inflammatory response in action. When you get an injury, mast cells release histamine, and macrophages release things called cytokines.
Okay, let's pause on cytokines. That's a term we're going to hear a lot. If the immune system has a language, that's it, right?
That's absolutely it. Cytokines are small proteins, and they are the critical signaling molecules. They coordinate everything.
So in inflammation, histamine is the first signal.
It is. It causes the nearby capillaries to dilate, which increases blood flow, giving you the heat and redness, and it makes them more permeable.
And that permeability is the key. It lets fluid and more immune cells flood into the area.
Right, a whole rush of phagocytic cells, mainly neutrophils, pour into that injured tissue. And that influx of fluid and cells is what causes the swelling.
And all that accumulated fluid, dead cells, and pathogen debris. That's what we call pus.
That is pus. But this can escalate. A really bad infection can trigger a systemic or body-wide inflammatory response. That can cause a fever, or in the worst case, septic shock.
A life-threatening condition.
A life-threatening drop in blood pressure, yeah. It's an overwhelming inflammatory reaction.
So this rapid response team also has molecular weapons floating around in the blood.
Two really crucial ones. First, interferons. When a cell gets infected by a virus, it sends out interferons as a warning to its neighbors.
Like an alarm bell.
Exactly. It tells the nearby cells to start producing substances that inhibit viral replication. And second, you have the complement system.
Which is about 30 different proteins.
30 proteins, yeah, just circulating inactively in the blood. But when they get activated on a pathogen's surface, they assemble into a complex that literally punches holes in the invading cell's membrane, causing it to burst.
It's an incredibly effective, broad defense, but it is generalized. So what happens when you need, the special operations team?
That's when you pivot to the adaptive immune system.
The one that's slower, but much more powerful.
It trades speed for incredible specificity. And most importantly, it has immunological memory.
And the soldiers of this system are the lymphocytes, the B cells and the T cells.
Right. They all start out in the bone marrow, but they mature in different places. B cells mature in the bone marrow.
B for bone.
And T cells mature in the thymus. T for thymus. And to get them to respond, they have to interact with an antigen. And the specific little part of the antigen that the receptor actually binds to is called the epitope.
Okay, let's take B cells first. They're the antibody factories. What do their receptors look like?
The B cell receptor is a Y-shaped molecule on the cell surface. When that B cell gets activated, it starts to differentiate into a plasma cell, which is a factory that just secretes a soluble version of that receptor.
Yeah.
That's what an antibody is.
And the great thing about B cells is they can bind to whole antigens, right? Just stuff floating around in the blood or lymph.
Yes, intact antigens. But T cells are much, much pickier. They operate under a far stricter protocol.
They can't just bind to anything floating by.
Absolutely not. T cells can only recognize little fragments of an antigen. And those fragments have to be presented to them on the surface of one of your own body cells.
And they're presented on a special molecule, the MHC.
The major histocompatibility complex molecule? Yes. This whole process is called antigen presentation. The host cell is basically holding up a sign that says, hey, look what I found inside me.
This is a really key point. There are two types of these MHC molecules, class I and class II. What's the difference?
So think of it as two different kinds of intelligence briefings. Class I MHC molecules are on almost all of your body's cells.
Okay.
If a cell gets infected with a virus, an internal threat, it chops up some of the viral proteins and displays the fragments on its class I MHC. It's a distress signal.
It's shouting, help, I'm infected.
Pretty much. It's a signal for the cytotoxic T cells to come and kill it. Now, class II MHC is different. It's only found on specialized antigen-presenting cells, like macrophages.
So the cells that go around eating things?
Exactly. They eat an external pathogen, break it down, and display the pieces on their class II MHC to signal the helper T cells. Class II is about showing the rest of the immune system what's been captured.
Okay, so we know these receptors are incredibly specific. We need millions of different ones for millions of potential pathogens, but we only have about 20,000 genes. This is the big aha moment, right? Gene rearrangement.
This is one of the most incredible things in all of biology. It's basically a molecular lottery. An antibody gene, for instance, isn't one solid piece of DNA. It's made of multiple segments, V segments, J segments.
Variable and joining.
Right. And during lymphocyte maturation, enzymes go in and randomly pick one V segment and one J segment, and they literally cut and paste them together, permanently deleting all the DNA in between.
So each individual lymphocyte is creating its own unique functional gene. It's physically altering its own DNA.
Exactly. That random combination happening across different chains is what generates the staggering diversity. Millions of unique receptors from a limited set of parts.
But if it's random, you're inevitably going to create some receptors that attack your own body. How does the system prevent self-destruction?
Through a process called self-tolerance. As these new B and T cells are maturing, they're tested. any cell whose receptor binds too strongly to the body's own molecules is eliminated.
It fails the test and is forced to undergo apoptosis.
It is. Only the ones that ignore self are allowed to graduate and enter circulation.
So once a non-self antigen comes along and binds to one of these specific validated lymphocytes, we get the massive response known as clonal selection.
Yes. The one lymphocyte that has the perfect receptor for that antigen is selected, and it's activated to proliferate, to clone itself thousands and thousands of times.
And that clone produces two types of cells.
Short-lived effector cells, like the plasma cells that make antibodies, and long-lived memory cells.
And those memory cells make all the difference. If you look at the data, the first time you're exposed to something, the primary response, it's slow. It peaks maybe 10 to 17 days later.
But the secondary immune response, the second time you see that same pathogen, it's incredibly fast and much stronger. It can peak in just 2 to 7 days. That's why vaccines work. You don't even know you've been re-exposed.
Okay, so once the right clone of cells is made, how is the actual fight coordinated? It seems like everything hinges on the helper T cells.
They are the orchestrators. The switchboard, really.
Yeah.
A helper T cell gets activated when it binds to an antigen fragment on a class II MHC molecule, so on an antigen-presenting cell.
The ones that show what they captured.
Exactly. Once that helper T cell is activated, it starts pumping out massive amounts of cytokines.
And those cytokine signals are the key that unlocks everything else. They're needed to activate both arms of the adaptive system.
Let's start the humoral response. This is the defense handled by antibodies floating around in your body fluids. Now, antibodies don't kill things directly. They mostly disable and mark.
So, for instance, neutralization.
Right. They physically glom onto viral proteins or toxins and block them from being able to damage your cells. Then there's opsonization, which is just a fancy word for flagging the pathogen, making it much more obvious and tasty for phagocytes.
And they can also activate the complement system we talked about earlier.
They can, which leads directly to the invader bursting. So that's the humoral response for threats outside your cells.
But for threats that are already inside your cells, you need the cell-mediated response.
And that's the job of the cytotoxic T cells. They're the ones that recognize those distress signals on the class I MHC molecules.
The ones on almost every cell.
The ones on almost every cell. And when a cytotoxic T cell finds an infected target, it doesn't eat it, it executes it.
It enforces cell death.
It does. It releases a protein called perforin, which pokes holes in the infected cell's membrane. And then it injects enzymes called granzymes, which enter through those holes and trigger rapid apoptosis. It's a clean kill that contains the infection.
The beautiful coordination of these two branches is really the entire basis of immunization.
It is. A vaccine just introduces harmless antigens to your body so you can go through that slow primary response and build up a population of memory cells.
So you're ready for the real thing.
So you're ready for a rapid, powerful secondary response if you ever encounter the real pathogen.
And we should probably clarify the difference between active and passive immunity.
Good point. Active immunity is what your body generates itself, either from an infection or a vaccine. You make your own memory cells. Passive immunity is when you're just given the antibodies, like a fetus from its mother or a snakebite victim getting antivenin. It's immediate, but it's temporary.
Of course, such a powerful system can sometimes malfunction, which leads to things like allergies.
Right. Allergies are just hypersensitive responses to harmless substances, allergens. When you're first exposed, your body makes a class of antibody called IgE, which sticks to mast cells.
And the next time you're exposed,
The allergen connects those IgE antibodies, and it triggers the mast cell to dump a massive amount of histamine and other inflammatory chemicals.
and in the worst cases, that can cause anaphylactic shock.
A dangerous system-wide reaction. But even more tragic is when that self-tolerance mechanism fails,
Autoimmune diseases.
where the immune system turns on the host, like type 1 diabetes, where it attacks the pancreas,
Yeah.
or rheumatoid arthritis attacking the joints.
And on the flip side of that, you have immunodeficiency, where the system is just broken.
Right, like SEID, where you essentially have no functional lymphocytes.
So to wrap this all up, we have to acknowledge that this isn't a static system. It's a constant evolutionary arms race. How do pathogens keep evading this defensive genius?
They're incredibly clever. A key strategy is antigenic variation. The flu virus is a master of this. It constantly mutates and changes the epitopes on its surface.
So your memory cells from last year's flu are suddenly useless?
They're useless. You have to mount a whole new primary response. That's why you need a new flu shot every year. Another trick is latency.
Just hiding out?
Exactly. Viruses like herpes simplex can go dormant inside your neurons. They stop making viral proteins, so there are no fragments to display on MHC. The T cells have no way of knowing they're there.
And then there's the ultimate sabotage, HIV, a virus that attacks the immune system itself.
It's diabolical. HIV infects and destroys the helper T cells. And because, as we said, helper T cells are the orchestrators,
If you take them out.
you cripple the entire adaptive response, both the humoral and the cell-mediated arms. That's what leads to AIDS, which leaves the body vulnerable to all sorts of other infections.
What a journey. I mean, from the generic pattern recognition of a toll-like receptor to a highly specific genetically engineered memory cell, the efficiency of this dual system is just staggering when you really think about it.
It is. The two systems rely on different forms of intelligence. Innate recognizes the generic threat and raises the alarm. Adaptive takes its time, but it brings overwhelming specific power and long-term memory to the fight, all coordinated by those crucial helper T cells.
So what does this deep dive into self-tolerance and genetic complexity imply for you, the listener?
Well, we talked about that mechanism of diversification, the random cutting and pasting of DNA in your B and T cells. Just think about that. The rule in biology is that every cell in your body is supposed to have the same DNA. But to gain the function of immunity, your body has to break that rule. It has to sacrifice the genetic integrity of these specific cells.
It's deliberate genetic loss to gain a function.
It's deliberate genetic loss to gain the power of specific survival. It really challenges our basic assumptions about the stability of our own genome and just underscores the incredible evolutionary lengths the body will go to in order to achieve the ultimate defense.